Okay, well, good evening and welcome to the Museum of Discovery and Science. Thank you for joining us here tonight for our Distinguished Speaker Series. My name is Brenda Bodiger. I am a programs manager here at the museum, and I will be the host for the evening. Due to the latitude and longitude of Florida here, we have the third largest coral reef system on the planet. This means that we have a very diverse marine ecosystem, and that also means that we attract a lot of world-renowned researchers and research to our state here. Save Our Seas Foundation, which graciously sponsors this series, funds over 300 projects in over 60 countries. We are lucky enough to have some of those world-renowned researchers here tonight, and they'll be talking about their various research in elasmobranch science. I'm going to introduce you to the researchers now, and then they will be giving a short presentation, which will be followed by a panel discussion, and then we would love to open it up to any questions that you might have. Our first speaker for the evening is Dr. Derek Burkholder. He is one of the Save Our Seas project leaders. He is also the director of Nova Southeastern University, Helmuth College of Natural Sciences and Oceanography Marine Environmental Education Center. Say that 10 times fast. He's also a research scientist with the Guy Harvey Research Institute and Save Our Seas Shark Center. He is also the principal investigator for the Broward County Sea Turtle Conservation Program. Dr. Marianne Porter, she is also a project leader for Save Our Seas Foundation. She is assistant professor of biological sciences at Florida Atlantic University. She is the founder of Florida Atlantic Biomechanics Lab. Her current research includes understanding shark skin stretchiness, lionfish spine puncture performance, and examining sharks swimming using 3D techniques in the wild. Dr. Stephen Kajiura is professor in the Department of Biological Sciences at Florida Atlantic University, He's conducted research for various agencies, including the National Science Foundation, the Department of Defense, and the National Marine Fisheries Service. He's also served as an elected member of the American Elasmobranch Society Board of Directors. Our final speaker for the evening will be Dr. Dean Grubbs. He's a scientific advisor to Save Our Seas Foundation. He's also research faculty and associate director of research at Florida State University in their Coastal and Marine Lab. He is member of the International Union for Conservation of Nature and a member of the NOAA Office of Protected Resources Small-Tooth Sawfish Recovery Team. He's immediate past president of the American Elasmobranch Society. So I'm going to hand the microphone off to Dr. Derek Burkholder, and please enjoy the evening, and I hope that you guys are thinking of some really great questions for the end. Thanks a lot.
Thank you all very much. There might be a presentation coming. There we go. All right. Well, it's very nice to see everybody out tonight. Thank you so much for coming. I'm very excited to be kind of kicking off this evening. I'm going to tell you a little bit about some of the research that we've got going on at Nova Southeastern University, both myself and also the work of Dr. Mahmoud Shivji, who is unfortunately was unable to be here tonight, but is director of the Save Our Seas Shark Center here in the U.S. So as Brenda was saying, I wear a couple of different hats at NOVA. I'm very fortunate to work with a number of different groups. Much of my research is focused on the conservation of both sharks and sea turtles. So today I'll tell you a little bit about just some of the shark work, but just in general, one of the things that many of the people in this room probably know is that as humans, we're pretty good at messing up things in our ecosystems, in our oceans, things like that. So, one of the things that is sort of a vein in much of the research going on in our labs is that our oceans are in trouble in many ways. Much of the work is going on. We do get a lot of things from our oceans. We get a lot of food, oxygen, all these great things. But in doing so, a lot of times we can do a fair bit of damage to these ecosystems that we're studying or using for ourselves. So, one of the things that we're focusing on tonight is really looking at some of these top predators. Things like the sharks and billfish. Many of these animals are threatened, endangered. Populations are going down. Not all of them, certainly, but there are many that are struggling. Some of these systems are, down over 90% from their historic values. One of the things about sharks in general is that many of these animals are at or near the tops of the food chains in these areas where they live. So they're very, very important in helping to sort of structure and balance many of these ecosystems where they are. So I am going to make it a little bit interactive tonight. Do you guys have any idea of some of the ways that we use sharks around the world? Get some hands? Yeah. Yeah, food. We do eat them. What else? Cosmetics. Yeah, we use some of the oils from their livers for makeups, lotions, things like that. What else? What's that? Yes, pet food, definitely. Anything else? Souvenirs, yeah. You go to some of these shops, you got your shark jaws on the wall, shark babies in a jar, all that kind of fun stuff. What else? Yeah, shark fin soup. That's a very big one. And so, yeah, we just listed a lot here, right? We've got food, souvenirs, trophies, cosmetics, all these different things, some medicines as well. So we use sharks for a lot of things around the world. Do you guys have any idea how many sharks we kill around the world every single year, on average? Get a couple of guesses.
Maybe like
250 million? All right, we got some ringers in the room. I see how it's going to be. Yeah, so we've got some big numbers here, and that's exactly right. We're looking at over 100 million sharks are killed every single year around the world. All right, just think about that number a little bit. 100 million sharks are being killed, and so it's very important to try to understand where these sharks fit into our ecosystems and how sort of the roles that they play. Maybe figure out ways that we can help conserve these animals a little bit more. As I said before, I'm fortunate to work with Dr. Mahmood Shivji at Nova Southeastern University. Much of his work for many, many years has been based around genetics and using genetic tools to study sharks. And so, when we look at something like this, where you've got thousands of shark fins laying out drying, in many cases it's very, very difficult to know what kind of shark those are. We don't know if that was an endangered species, a population that's doing well, what's going on with that shark fin trade. So what Dr. Shivji's been doing is using genetic tools to try to track the shark fin trade a little bit. And so by taking a little piece of this fin, run it through the machine, using genetic markers, he's been able to develop some different forensic tools using these genetics to try to understand a little bit better about what species are being found in the shark fin trade. This really has opened a lot of eyes to kind of what's been going on, and really was one of the first places where a number was actually put on how many sharks, and that was where I heard 73 million over here. His work estimated that up to 73 million sharks were being killed strictly for the shark fin trade, or at least at the time when that paper was being done. And so it's very interesting to see kind of what is going on around the world, where these species are where these sharks are coming from, what species are being taken, things like that. Much of the work that I've been doing is really focused much more in the field. So there's some amazing tools and technologies that allow us to learn a lot more about sharks. You know, these animals live their entire lives underwater, so for us to be able to go out, you might be able to dive or spend a little bit of time with them, which is fantastic, but as soon as they swim away, that's when it gets a little bit trickier. So to figure out a little bit more about these animals, a lot of times we go to different technologies and different tools to try to learn a little bit more about them. One of the tools we've been using quite a bit with the Guy Harvey Research Institute at NOVA are satellite tags. So satellite tags are an electronic tag that we mount on the fin of a shark, send that shark out into the middle of the ocean, and anytime the fin breaks the surface, that tag's going to ping, send a signal out, ping a satellite, send us a notification of where that shark is anywhere in the world. So these are fantastic tools that allow us to look at much more long-term movements of what these guys are doing. So we've been working with many different shark species. A lot of our recent work has been focusing on mako sharks. Makos are one of those highly sought after, highly caught for their, as a sport fish. They're a lot of fun to catch. Jump, flip, flop, all that kind of fun stuff. So they're a lot of fun to catch. And so much of our work has been focusing on what these animals are doing once they leave the side of the boat again. What we see here, each of these dots on the map here all represent a surface where that shark came up to the surface of the water and sent out a signal telling us where that shark was. And so each one of those dots is a location from this satellite tag. So you can see these guys are moving pretty incredible distances. Some of our makos are traveling up to 15,000 miles a year, and that's just measuring sort of the point to point at the surface where we're looking at them. And when we talk about sort of conservation and management of things like sharks, billfish, tunas, these highly migratory species, these tracks really start to open our eyes a little bit. If you look at the different colors on this map over here, the different shaded areas represent the exclusive economic zones for these different countries, the US, Bermuda, Canada, Mexico. And with this particular shark, traveled through at least 4 different management zones over the course of a year. So every one of those is going to have their own set of rules, their own set of regulations. We've had some of our makos travel through as many as 18 different countries' waters over the course of a year. And so it really does open our eyes to sort of the struggles that we face as a community to try to manage and accurately protect these guys a little bit more in all these areas where they're traveling. I will show you just a couple of these tracks here, hopefully. Maybe not. All right, let's see what these work. All right, so what we're looking at here, this is a tracking website that we've put together to kind of show what these animals are doing. So when you first pull it up, you kind of take a quick look at some of the different species we've been working with. Tiger sharks, makos, oceanic whitetips, recently working a little bit more with things like whale sharks, and even some of the billfishes, marlin, sailfish. And so one of the first projects that Dr. Shivji was working on is they wanted to travel to Bermuda, a little tiny island out in the middle of the Atlantic Ocean, and figure out what the tiger sharks might be doing there. So they put a few of these tags out. We come over to the side over here. These are all different sharks that we've tracked. And again, each one of these dots represents a place where that shark came to the surface. If we take a look at this, if you look at the top there, we've got a date code. So this was tagged in September. Pretty quickly made us a beeline down to the Caribbean. It took about a month to get there, but got a little bit cold up north, went straight to the Caribbean, and is going to winter down here in the Caribbean islands. Pretty smart shark, I feel. Nice warm water, lots of food for him. He's going to spend the winter down there. As it starts to warm up in May, June, July, the shark travels out into the middle of the Atlantic Ocean. It's going to spend the next couple of months bouncing around out here, maybe foraging, maybe mating. We don't really know what's going on. But then we're going to see a little bit later is going to kind of make that full circle. And what's pretty incredible to me is these guys, they don't have the iPhone in their pocket. They don't have a map that they're reading telling them where to go. But 1 year later, the next winter is back in the exact same set of islands in the Caribbean. overwintering again, hanging out, and then as it warms up, it's following this same path over and over and over again. This particular animal, we were very, very fortunate and able to track for over 3 years and follow this same pattern over and over and over again. Winter down in the islands, summer out in the middle of the Atlantic. You can see it looks like it's taking a little bit of a different path. That's just because it came to the surface a few more times on that particular pass. But what this shows is that, you know, these animals are moving, at least in many cases, in a pretty determined pattern. You know, they're going same places over and over and over again, whether it's following their food or temperatures. Those are the things that we're still, in many cases, still trying to figure out. But it does show just the massive movements that these animals are moving. As I said before, we're doing a lot of work right now with the mako sharks. And so if we take a look at this, one of the things with this website is that, and this is live, you can look at anytime, this is ghritracking.org. Anytime you see the little Wi-Fi blinky green signal over here, if we click on that, this is one we've heard from in the last 2 weeks. So as I said before, anytime they hit the surface, ping a satellite, tell us what's going on. So this particular animal, that shark fin is where that shark was yesterday. So as we're learning, this stuff goes on the website and goes out, and you guys can kind of track and follow what these guys are doing. You can see over on the side here, all these different sharks that we've tracked. There's a lot of red dots, unfortunately. These are animals that have been caught and killed by fishermen after they were tracked. And so they were moving around doing their thing, and at some point, you know, this guy didn't make it all that far, got picked up here, taken to shore. Same here, this guy was cruising around, was caught by fishermen and landed over here. These are all legal catches, but unfortunately we're showing a pretty high rate of capture with these animals. And so in 2017, Dr. Shivji and his team were able to publish a paper looking at these makos. And so with these satellite tags, we can see a lot of great information about their movements, what they're doing. But one of sort of the side notes that came out of this is that at the time, all the management that was done with these animals was based on about a 3% capture rate of the population estimate. Of our tag sharks, over 30% were being caught and killed, usually in less than 6 months of being tagged. And so we're looking at a much, much higher rate potentially of take than what was estimated previously. This has opened up a lot of doors, and there's been a big push now to try to list these and protect them a little bit more. One of my projects here, and something that I've been very excited about and very fortunate to work with for a number of years now, is bringing some of this shark work right here to South Florida. And so with this project, we're really just trying to understand what shark species are here, look at their seasonalities, what they're eating, those kinds of things. And in doing so, every trip that we go, we want to make sure it's an educational experience. And so every trip we take, we bring a group of people with us. Most of these are middle school, high school classrooms. We're fortunate to work with Pompano Dive Center. We take out, they're nice big dive boats. We can take up to 30 people at a time. We get people out and get them hands-on. And so this is something it's a lot of fun for me personally to be able to share this research and my passion for the oceans and conservation and sharks, but to be able to get people out and see and hopefully break a few of those stigmas of what sharks really are as they are able to get hands-on and see a little bit more about what's going on. So this is something that is open to the public, so if anybody at the end of the day here is interested in joining on a trip like this, please come find me. I would love to talk to you about it, or if you have a school group or anything like that, we'd love to get you involved with this trip. So with that, I would like to take maybe a question or two. I know we're going to have the panel later, but if there's anybody who has any questions right now. So he's asking, how long can the satellite tags last if the sharks aren't caught or killed or whatever? It depends on a whole suite of different things. It depends on how often they're coming to the surface. The tags are designed so that when they're underwater, they're shut down, so they're not burning battery, and they just wake up and ping at the surface. So generally, it's usually going to be something other than battery life, usually that's going to stop them. Some of our longest tracks were a little over 3 years. Most of them we get about a year, a little over a year maybe on a satellite track, but we have had some quite a bit longer. We've had some 2 or 3 days. So it just depends. At the end of the day, we're putting a little computer on the back of a shark and throwing it in the middle of the ocean. So there's a few different things that can go wrong with that, unfortunately.
How do you tell if the shark got caught by a human or just got killed by natural causes or predator?
All right. So Dr. Grubbs might be able to speak a little bit more about the potentially killed by something else later, but in our instance, what we found with these satellite tracks is that they were moving along, they were out in the middle of the ocean, and then the next day they were onshore, and in some cases we were even able to zoom in. These kind of just plot on Google Earth. We were able to zoom in and find what dock it was sitting on, and in some cases those were commercial fishing operations. We were able to call them up and they'd mail us the tag back. So usually it was just that it ended up onshore somewhere. Many cases, if they would die of natural causes, we would never hear from them because the shark would usually not come to the surface. They'd usually sink or something like that. Great question. Yeah.
What's one of the hardest sharks to have?
One of the hardest sharks to tag. They all have their own unique challenges, I would say. For me, one of the hardest ones that I've worked with, and again, Dr. Grubbs has done a lot of work with these guys, are the sawfish, and more of the ray species in the ray families, but they do provide some unique challenges with the saw coming out the front there and being able to restrain, in some cases, very large animals to be able to tag those. So as far as my experience, that was one of the hardest ones I've dealt with for sure. So.
What else do you guys track besides location?
What's that?
What more data do you guys track besides location?
So it depends a little bit on the tag. So she's asking what other data do we collect besides location. In many cases, these spot tags are largely collecting just position, but there are some pop-off tags that are also collecting depth and water temperature, things like that. There's other iterations of these tags that may have accelerometers built in that are giving very fine scale, three-dimensional movements, 3-axis movements of exactly what they're doing. But for most of the tags, it's giving us largely position and temperature. All right. So I think we're going to move on. Thank you all very much.
All right, we'll see how this works. I actually have lots of movies in this presentation for you. So with more technology, there's more fun and exciting things that can go wrong. So while Dr. Burkholder can tag a shark and tell you where it moves on the planet Earth, I study biomechanics. I like to think about how that actual shark is moving. So while it may go from place A to B, I want to know how it gets there. So this is an entire field of science called biomechanics. So we use biology and engineering and physics to think about how these animals move. And so we're applying mechanical principles to biological systems and thinking about form and function. So how are these animals shaped and how are they getting from where they're going? So we like to think of mechanics, how it works. So any of you who loved to play Legos when you were kids, building stuff, putting it together, that's basically just my job now. So if you like doing that, that's great. And it turns out, not only is this something that kids are interested in, it's something that we humans have been interested in for thousands of years. So Aristotle was working on books called On the Movement of Animals, 300 BC, and Giovanni Borelli and Leonardo da Vinci were making these amazing drawings of human lever systems of our bones hundreds of years ago. So we've been interested in this field for a very long time. I'm really interested in the cartilaginous skeletons of sharks and how sharks move around. And why is this cool? Well, every other vertebrate animal is going to have a skeleton made of bone. We have bone, sharks have cartilage. Now imagine if you were to go running, if you wanted to. You could go outside and go, I wouldn't, it's too hot. You can go outside and go for a run. Your skeleton's going to hold you up and handle the forces you're putting on it. Now imagine your skeleton is made of the squishy stuff in your nose or in your ear. What do you think happens if you're a very athletic animal when you do that? And like the mako sharks Dr. Burkholder was talking about, they're some of the fastest swimming fish in the sea. And so they're very athletic, and they can do these amazing feats with skeletons made entirely of cartilage. So some of the big questions that drive my research are, how does this cartilage work as a mechanical system? And better yet, when you put this mechanical system in a shark, how does the shark actually move around? And so today I'm going to show you some fun things in the work that my lab does. So we're the Florida Atlantic Biomechanics Lab, the FAB lab, and our goal is to study marine organisms and the diversity of marine organisms, thinking about how they work as mechanical systems. And so here, just to take a look, this is what I like to do with my life. It's just watch sharks swimming. So here's some video that Dr. Kajiura took, actually, of some blacktip sharks swimming around. And we can see them swim. That one looks like it moved its pectoral fin to maneuver. They wiggle their bodies to swim around. And so how do we actually study this as scientists? And so one of the things we can do is we can study how these animals move in 2D. We can catch them, we can bring them back to the lab, and we can put cameras over their tanks and watch how they swim around. This is what we call volitional swimming. It studies how sharks move of their own free will. One of the other ways people in my field can study how sharks swim is they put sharks in a flume. It's an underwater treadmill. And I actually prefer this way of studying how sharks swim because the shark gets to pick its swimming speed. How many of you have ever run on a treadmill? Most people. How many people let someone else pick the speed that they run on the treadmill? Not very many people are admitting to that. Why? I guess it's like feels really weird. If I were to go to the gym with all of you and picked how fast you were going on a treadmill, I might pick something too fast or something too slow. And it might not be the way that you prefer to actually move. And so I like to study sharks swimming sort of in large tanks as they swim, so I could really get a feel for how they prefer to actually move their bodies and what speeds they prefer to swim. And so what we do is we put them in a tank and we're able to videotape them and then look at specific points on their body. If you think about this animal, there are millions of points. Every single space on that body I could track and try to understand how this swims. But I mostly like to look at, oops, some of my slides are gone. We'll just keep doing that. Oh, my video stopped. Okay, there we go. Technology, right? So we can put specific points on these bodies, on the midline, on the eyes, on the cephalofoils of these hammerheads, and we can decompose this body into stick figures and really think about how the animals are moving around. So some of the things we can look at are how fast they beat their tails, how far back and forth they beat their tails, and how their body wiggles. And we're able to learn things about how these animals swim. So one of our recent studies, we were able to look at these hammerheads and then also bonnet heads, so the species of hammerhead with the smallest hammer, and we were able to show that while they swam the same speeds, they actually wiggled their bodies in different ways to kind of achieve that overall output. And so why is this important? Because we're starting to understand how these animals work. So we can understand where they go, how do they get what, or are they going from A to B, but how did they get there? These are important questions in terms of the basic science and basic physiology of these animals that are important to consider as we think about how to conserve them. Another thing we're doing is really trying to understand how animals move in 3D. 2D, I can get that nice top-down view of the shark and I can see how its body wiggles. But if you've ever been out diving or at an aquarium and you've watched fish maneuver, they use their fins an awful lot to do things, right? And I can't see that movement when I'm just looking at them from above. So what tricks do we have? Well, we need to start thinking about things in 3D so we can look at them from the top and from the sides. And so for this project, one of my graduate students and I were innovating how to do 3D underwater videography to really understand how sharks are maneuvering using their pectoral fins. Why anyone would care, you ask? Well, we're interested in developing underwater AUVs, so underwater vehicles. Airplanes actually move around in fluids, right? Air is a fluid. So we can begin to understand how animals are moving their fins to make careful, fine maneuvers. And we can use that information to not only have a better idea about shark physiology, but also use that for bio-inspired designs that we can use for humans as well. And so what we were able to do to start to study animals in 3D is very similar to what we see when we see the making of movies. So if you ever watch to the very end of a movie on the DVD, they always have how they made the movie, right? And so if they're using 3D technology, we see things where we have actors in the little suits with the dots on them, and we want to be able to decompose their very complex movements, again, into these specific points. And so we can turn them into a cloud of points and then make them little stick figures. So we've gone from essentially millions of points to a couple hundred, maybe 1,000 points, and we've got these stick figures that then we can animate and start to make our movies. And we've used this kind of technology in biomechanics for a long time, but one of the challenges is being able to take things underwater. Life becomes more complicated when you take robots or electronics or anything underwater. And so what we have been able to do is actually take this technology underwater. And we've been able to do this in a way that is actually accessible to researchers around the world. So we've been able to do these experiments using GoPro cameras, so commercially available GoPros. How many people here have GoPros? Like everybody, right? And we use open source software that's freely available to any researcher around the world. So the papers we've been publishing on this, researchers anywhere, regardless of how much money they have or the resources they have available to them, all they need is water, some rocks to put on the bottom of a tank or wherever they're looking, their sharks, and some GoPro cameras, and they can do these experiments. So this is a really great project in terms of moving our understanding forward, but also inclusivity and open access to the scientific community. So what we were able to do is create a system where we have our little rocks here, creating basically the flat surface of a square, and that creates a cube in this water here. So imagine if each of these rocks were to have a line going up. That would be our cube. That's our volume that we're interested in. And then we basically take a giant scaffolded structure that was made out of Connects. So again, freely available open source stuff you can get, Connects. And we were able to calibrate every little bit of this square, this cube. And then we have our GoPro's cameras out here, so 2 of them that were kind of 45 degrees to each other, so that we can see from a camera all of this volume. We were able to put little dots on our shark. These are little beads that have been painted black, and they're glued to the shark. Now everybody always asks, well, doesn't that bother the shark? Well, how many of you have petted a shark in like a touch tank or something? Whole bunch of people. What's it feel like? Sandpaper. And that's because shark skins have these little tiny dermadenticles. It's essentially little teeth all over their skin. And so when we glue this on, it comes off by the time our experiments are over. That was actually how we knew our experiment was over, as we started losing all of our beads, because it just falls off because of their skin being so rough. So what we did was we were able to paint all these little dots onto our shark, convince it to turn through this area, and now we've got something no one's ever been able to look at before, the 3D maneuvering of these bonnethead sharks. And so next, I'm going to show you some pictures of a dogfish shark. These are experiments we did with another species. We started off with something a little bit smaller. So we have our dots. We draw the lines between it, just like we do in the movies. And then we're able to use this open source software to start making our animations. So now you can see the shark from the movie swimming in the background, or the shark from our data that we got with our GoPros, and some of our animation. We can take away the shark and the GoPro movie, and now all we have left is our animation. So we've greatly reduced the complexity of our shark, and we're able to now hone in onto those movements that we're very interested in, the pectoral fin. And then we can hold our shark still and actually make measurements of how this pectoral fin is moving. And from our research, we've been able to show that this little dogfish shark is actually moving its fin in such a way that it slows down through turns, whereas the bonnethead shark moves its fin and accelerates through turns. And we have the ability to look at different types of maneuvers. So with the bonnet head, we were able to show that some of its turns, it would go put its little fin down on the tank and sort of rotate it around it like a speed skater does as it's going through and accelerating through its turns. So that's some of the work we're doing in 3D. What happens if you want to do something outside? So I've been working with sharks in the lab, thinking very carefully about studying how they swim, not just putting them on the treadmill, right? But ideally, we would be able to look at them outside and think about how they're swimming actually in the wild. And it turns out we have a really great opportunity to do this. I'm sure Dr. Kajiura will talk a little bit about the blacktip shark migration after this. But we have all these sharks that migrate down here and they hang out here. I don't want to steal any thunder that's coming next. But we have these sharks. They're in nice, shallow, clear water. Very similar to the water that you saw in my tanks, right? We've got shallow water. The bottom is kind of this light blue. We can see the shark's dark body there. We're able to take our little drone here in red. I send my grad student to the beach with a drone. He goes to the beach with a drone, and this is his PhD research, Braden Ruddy. And he gets video of sharks swimming and maneuvering. So this is actually kind of a rough day from some of his video, but we see sharks swimming around. And this is gorgeous video, right? If you remember back to that head overview video of the hammerhead swimming in the lab, this is kind of comparable. So we're able to use many of the same techniques that I've been developing over the years in the lab to study how sharks are swimming, and swimming volitionally, right? Not in that treadmill. And now we're able to have a species where we can start to look at this in the wild. And so, again, here's some lovely video of sharks swimming straight through. This is exactly what I would want to see in the lab, but now I can look at this and examine it in the wild. And not only that, we can look at how these sharks swim by themselves, and we've been able to get great video for groups of 12 sharks swimming together, so we can start to understand how they move together in a collective way and start to analyze their behavior of how they're moving around together. And then we've also been analyzing some video where we look at predator-prey interaction. So there's some video where the hammerheads that prey on this species come in. So now we can get the swimming and understand how the blacktip shark is moving, but we can also understand how the hammerhead is moving in a predatory event, which is really exciting and things that we had never been able to look at before when we were studying how fish swim. So again, we can slow these video down to the frames, track the point just like we've done in the lab, and we can have our shark and learn a whole lot about how it's swimming. So we know it goes from point A to point B, but now we're starting to really understand how they do it. And again, my lab does a lot of work in bio-inspired designs. So we've got some fun work going on with lionfish, which is, as you know, an important invasive species locally, thinking about how shark skin works and how the vertebrae, that cartilage in the shark skeleton, is working as mechanical systems. And then with that, I will take any questions or wait till we have our next time, our panel time. Oh, the biggest surprise that I found? Well, one of the things I've learned as a scientist is almost every hypothesis I have about my projects turns out to be wrong. So there have been lots of surprises. But the most exciting thing that I found so far is about the shark skeletons, their backbones. So when we move with our bony skeletons, we move, our bones move around our joints, right? But it turns out the shark cartilage can squish just a little bit. So as they're swimming, the joints in their vertebral column are moving, but their actual vertebrae, like the bones in their skeleton, are moving as much as the joints. I was totally shocked and measured it like 10 times because I thought I was wrong. So it's really great because it shows that the vertebral column the whole thing is actually able to smush and move, which creates what we call a biological spring. And springs, if you think way back to when you took physics, are important because they can store and return elastic energy. So if you're thinking about animals that make long migrations, having a springy body would be really useful to you. So that was like the most exciting and surprising thing. And like I said, I had to remeasure it a bunch of times because I was like, what did I do? There's a number missing here. Yeah, any other questions? All right. Oh, yeah.
Do you see any differences in the age of the animal, like the way they move, like the
Question is if I see differences in movement based on the age of the animal. So the way we can get at that is studying the shape of the animal. So one of our projects right now is looking at little epaulet sharks, which are a shark from the Indo-Pacific that can walk. So they walk from tide pool to tide pool. And we did a project looking at the neonates that still have an internalized yolk sac, so they're really round and bulbous, compared to the juveniles that were more slender. And it turns out that they were moving at the same speed and they were moving their feet in the same way. And so there didn't seem to be much of a difference in that respect. But many of the other things we do, there's just simply not enough data to actually say anything about it. So yeah, it's a good question. Yeah.
Talking about sharks, studying sharks inside tanks. I've heard that they've never been able to keep a great white inside a tank to study them. Have they ever found out why?
Oh, that's probably a sensory physiology question. Dr. Kajiura can say that. But at the Monterey Bay Aquarium, when I was in California doing my PhD, they had a baby white shark that they successfully kept in the tank for a while. And I think they did that with several sharks, and they were able to keep them until they reached a certain size. Yeah, Dr. Grubbs is like, yeah. So they were able to reach a certain size, and then they started getting, I think, more aggressive to the soup fin sharks that were in the tank with them, I believe. And they just started getting bigger. I think it's a space issue, probably more than anything else. You have these animals that they live, in the ocean, and you put them in this little tank. It'd be a little disconcerting, right? Yeah.
When you were talking about the sharks and the groups, if you found any interesting or unexpected observations with regards to the group dynamics, and if so, what were they?
Yeah, so that's a great question. So a lot of what we know about how animals move in groups are from animals that we can see on land. And there's some work starting to think about how fish swim together. And there's a whole lot of theory that actually comes from physics theory, thinking about how particles move together. And what the theory tells us is animals should move in like a little diamond shape, and they should get benefits from kind of drafting behind the animal behind them. And we haven't really been able to show that very often in the wild. It's been shown in some experimental work with smaller fish in labs, but we haven't been able to show that. And what we've been able to show so far with our drone video is it does seem like they form these groups of four. So that was really exciting. My grad student, when he found that, was like, look. And then I'm like, great, it happened once. And then he came back like 2 months later and was like, look, it happens more. So yeah, that was really exciting. Anytime we have theories and things that we think should happen, and we have these hypotheses. So when we are able to actually go out into the real world and observe it and actually measure it, and we're like, yes, that is how it works. It, of course, opens up about 50 million new questions for us, but it's a great thing. So that was exciting to actually show that that is happening in sharks. And one of the things we're doing with that project is working with an engineer who studies hydrodynamics. And so all of his work is based on models. And he was saying it was really great, and I really wish I could tell you more, but we just don't know how real fish swim. And so we showed him our videos, and we're like, we have numbers on how the real fish swim. So we're actually plugging the numbers from our sharks that we've been able to measure into his models to start to make computer models of the hydrodynamics of how we think the water is moving behind these sharks, so we can start to really get at what kind of benefits they may have with that sort of drafting behavior. Cool. All right. Do more later? All right. Thank you.
Okay, our next presenter is Dr. Stephen Kajiura. He's professor in the Department of Biological Sciences at Florida Atlantic University.
I'm going to talk to you about some of the work that we've been doing looking at the migration of the blacktip sharks down here. Now, this sort of work is not my forte. My training is actually as a sensory physiologist. I work on things like electroreception, how the sharks detect electric fields in the environment. But about 10 years ago, I started to get involved with these blacktips that would come here to South Florida. And what would happen is the news helicopters would go along and they'd see all these sharks here and they'd give me a call and say, what's going on? Should we be concerned? Why are there so many sharks here? And I said, well, it's the same thing I told you last year. They come down here, they spend the winter, and then they go away again. And so people were concerned that they were seeing things like sharks close to the beach, and you can see literally hundreds of sharks right up against the beach. There's some people right there too, right, with the sharks, all right? So you get an idea of how close these sharks are down here in South Florida to the beach. And guys could be walking along the beach, literally walking along the beach, and there's like a dozen sharks right there as well. And so this is what was getting a lot of attention, especially for some people that have really nice houses who happen to be right on the beach, who have sharks right in their backyard. And so I was called in and said, what's going on? I said, I don't know. It's basically this migration. Let's find out. And so I started to do an aerial survey. I figured all the best data was coming from these news helicopters anyway. Let's get up in the sky. Let's see what we can see. Let's see the sort of bird's eye view and get an idea. And so I started by taking a plane, just a little Cessna 172, and I would mount a high-definition video camera out the window of the plane and also a still camera behind it. And we fly transects. So for the first few years, we just covered Palm Beach County, from Boca Raton up to Jupiter. And then subsequently, we covered the entire Tri-County area, from Miami all the way up to Jupiter. That's to 2019 now. And we would just fly along the coast here with the cameras mounted out the window of the plane. And this would enable us to record this stretch from the beach to about 200 meters offshore. And record it all, take the video back to the lab where we could carefully go through it and count the numbers of sharks. How many were there? Where were they distributed along the coast? What time of year were they here in greatest numbers? And get an idea. So I was able to answer some of these reporter questions. And what we saw, this is the sort of cartoon view of how the setup is run. There's the airplane. It's flying out of the screen towards you. There's the beach over here with the little umbrella and the water with all the sharks in it. And what the camera is seeing, it has a field of view that covered from the beach to about 200 meters offshore. Now the nice thing about that is the water down here is pretty shallow and nice and clear. So even at 200 meters offshore, you're only at about 4 meters deep, which means that you can see all the way to the bottom. You can see the sharks silhouetted against a nice light sandy bottom very easily. And that made it easy for us to see them, even though we're up in an airplane flying 500 feet above them. And so this is what the camera would see. All right, on the left side of the screen here, this is the beach. All right, that's the strut of the plane. There's the wheel. And every one of those little black dots is a shark. And so you get the idea that that's a lot of sharks to count. And if you've got literally 2 seconds as you fly past this thing at 70 knots, how many sharks are there? Go. 6, how many do you think? 236. Anyone else? Give me a number. 5,000. And give me another number. Okay, 1,500. That's good. Who said 1,500? You say 1,500? No, you should have. There's 1,678 sharks in that frame alone. All right? And just to give you an idea what this looks like, I'm going to show you the clip from which I extracted this. It's just a 30-second video clip from the aerial survey. What the camera is seeing as we're flying along. And we're in an airplane. We can't go any slower than this. But you see all the sharks? Look at them all. Those are all sharks all along the beach here. Literally hundreds of them. And this is why you have to go with the video camera and go back after the fact. You can't count this in real time. There's just too many. And then you get these massive aggregations like this. Literally thousands of sharks just passed beneath us as we're flying along. And it just keeps going. I mean, it doesn't stop. There's more. I mean, it goes on and on like this. And like I said, this is just 30 seconds out of the whole transect along the coast. But I do want to point out a couple of things. Number one, there's lots, as you can see. Number two, they're close to the beach. And number 3, there's the road. There's A1A right there. You can literally drive down the road, Ocean Boulevard, and you could throw a rock and hit a shark. Don't do it, but you could do it, right? It's possible. They're literally that close to the beach. And so what we're able to do is start counting. Go in and take the video footage and look at how many sharks do we see. And from the time we started, we were seeing in excess of 12,000 sharks on a run, and the number would drop off precipitously in the spring. So by April, pretty much nothing. Throughout the whole rest of the year until the following year, jump back up again. For a couple of months, you had huge numbers of sharks, and it dropped off. This year was a bit unusual. 2013, we had a lot of this beach renourishment. You know what this is? When you pump the sand from offshore onto the beach to make the beach bigger and wider. What it does is it sets up an enormous silt cloud so you can't see anything. So there you got, if there were sharks there, you couldn't see them. Or what might be happening is the sharks might be going farther offshore with outside the transect. Either way, the numbers were really low that year because of this. But they bounced back up again the next year again. Now, if you look, you can also break this down. I'm going to come back to that figure in a second, but I'm going to break this down a little bit. We're interested in the distribution of these sharks along the coast as well. Are they all in one area? Are they evenly spread? Are there concentrations? And so we could break the transects up by inlets. You can see these inlets as you're flying along. It's easy to see from the plane. And it made natural little sections along the coast. And we're able to say we know the distance between each of these inlets. You can go just in Google Earth and measure that. And since we know the width of that transect, it's about 200 meters, right? The length of 2 football fields out from shore. 200 meters, we're able to say, given that transect width of 200 meters and given the length, we're able to get the area. How many square kilometers were we sampling in each of these little sections here? And then we can count the numbers of sharks in each section, all right? How many sharks in this little transect? And we're able to say there are X number of sharks per 3.9 square kilometers to get a density. How many sharks per square kilometer were we actually seeing? And that's where we would get some interesting results. In the wintertime, before the sharks got here in December, right? Sharks weren't getting here until January, February. So we would run transects in December to see who was there as our baseline. And we would average maybe 0 sharks, 1 shark per square kilometer, maybe 6 on occasion on this one time by Boca, but not very many. You contrast that to just a few weeks later, during peak season when the sharks are here in February, and now the densities change dramatically. You're not talking 1 shark per square kilometer. Now you're in excess of 1,200 sharks per square kilometer. And a lot of the sharks were up north, from Boynton up to Jupiter. Not as many down south, a few around Miami, but not nearly as many as we saw up here. And just to put this into perspective, what does 1,231 sharks per square kilometer mean? It's a lot, but how do you wrap your head around that? So what I tell people is, think of it this way. Go to the beach, and next time you're swimming in the water, 1,231 sharks per square kilometer means that if you draw a 16-meter circle around yourself, there's a shark there. Doesn't necessarily mean there will be, but there's the probability. That's how dense we're talking here. So next time you go to the beach, take an imaginary circle, draw it around yourself like this, and imagine somewhere in that circle, there's probably a shark. All right? At least during peak season. Go now, there's nothing. There's hardly anything out there. All right? All right. So huge peaks followed by nothing throughout the summer months. Pretty much nothing throughout the summer months. We found that there's a really strong correlation with temperature. Lots of sharks when the temperature is really low. Water temperature is really high, hardly any sharks. So it's this inverse up and down relationship between lots of sharks with low temperature, high temperatures, very few sharks. And so the sharks have this maybe preference for a certain range of temperatures. And you can see this by looking at when we're seeing the sharks. We're seeing sharks in big numbers, lots of sharks, only when the temperature is low. The temperature is less than 25 C. Temperature is warmer, we only see a few sharks here and there. Okay, so sharks have this strong correlation with water temperature. All right, stick that in the back of your head for a second. If you look now at what the mean water temperature has been doing here for the past 8 years, mean water temperature has been creeping up over time. All right, that's a significant slope, believe it or not. And what do you think has been happening to shark numbers then? It's the opposite, right? Shark numbers have been going down, 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 down. And in fact, if you looked at 2018, that was the smallest number of sharks that we had ever seen. 2017 and 2018 were the two worst years that we've ever had on record. And 2017 was also very warm in terms of temperature. 2018 had a very brief cold spell for about 3 days in January, which drove the mean down. But if you throw out those 3 days, it's way up here again. So 2018 was also warm. And so you've got this strong correlation between temperature and shark abundance. We're going to talk about global warming in a little while and what impact that's going to have on shark distributions. But just stick this in the back of your head for now. All right? Moving on. The next thing that we do to study sharks from the sky, in addition to flying airplanes, is we take up drones, like you saw Dr. Porter talking about. Now, the nice thing about drones is they're inexpensive, they're easy to fly. You can literally walk down to the beach like she was saying her student does. And because these blacktips occur so close to shore, it's easy to take advantage of them by flying your drone right over the sharks and getting some really compelling footage. We use this for a couple of reasons. Number one, it gives you that wow factor. When you see a dense aggregation of literally thousands of blacktip sharks like this, you say, wow. Come on. Wow. Thank you. All right? It's pretty impressive when you see that many animals out there. And this enables us to look at things like social behavior. How are these sharks interacting with each other, for example? Are they all forming like a polarized school, swimming together? Are they all off doing their own thing? These sorts of behaviors we're able to address by getting this top-down view. The other nice thing about drones is it gives us a real advantage when it comes to fishing. We're able to fly the drone, see where the sharks are, and then say, well, that's where we're going to go fishing then. And cheat, basically. Throw our bait right where the sharks are going to be, or right where the sharks were. And so there's definitely advantages to drones when it comes to capturing the animals. And then finally, it gives us the ability to look at interactions between different species. So we're looking at the blacktips, but we're also interested in the predators of the blacktips, the hammerhead sharks. And so here you have a hammerhead shark cruising along looking for a meal in one of these blacktips, and just watch really carefully. This hammerhead is stealthily sneaking up on these guys, and look at how close he gets before the blacktips even realize that that hammerhead is there. Once they realize it, of course, boom, and of course everyone goes away, and poor hammerhead's left with nothing. Yeah, he tries really hard. But again, we're able to get this sort of natural history, these sorts of behaviors that are hard to get from the water eye view, which you can get when you're doing this sort of top-down drone view. He tries again, but of course the blacktips are a little more wary now, so they're not going to let him get nearly as close. And I'll let you watch this for just a second because it's fun. But he tries to track him down one more time. They're not having any part of it, but it's pretty neat to watch these sort of natural predation events. You know, predation is relatively rare to see in the wild, and especially rare to see for organisms that are underwater. So to have this opportunity to view this sort of natural predation is, I think we're really fortunate to have clear water, shallow water, where we have lots of sharks, both predators and prey, that we're able to document this and actually study it scientifically. This is the footage that we're providing, with Dr. Porter's lab. Look at that, boom, 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 and I cut it off before you see anything bad. You don't know what the audience is like. All right, so I just want to end by pointing out that there's a lot of people out there on the water, there's lots of sharks out there on the water, and we have relatively few interactions down here in South Florida. I think we're very fortunate that we have nice clear water. People are able to see the sharks are able to see the people, and the two kind of stay away from each other. And so we have relatively few negative interactions, let's say. And I think we're fortunate down here to be in South Florida. We have these environmental conditions. So I'm just going to wrap it up by acknowledging our funding which is coming from the Colgan Foundation. And I encourage you, if you have your phones, pull out your phones, follow us on Twitter, Instagram at Shark Migration, and our Facebook page is FAU Shark Migration. So I'm going to wrap it up with that, and if you, I'm not sure if we're taking questions now, if we're holding out till later, but if you do, I'll take 2 quick questions right now. Thanks so much. Yes, sir.
Steve, I just wondered if there's any kind of communication between, when the hammerhead's coming in,
Right.
and the sharks communicate to each other that there's a danger.
Right, so the question is there any sort of communication between the sharks, any sort of social interaction where the sharks are saying, hey, watch out, there's a hammerhead around here? We have no documentation that these sharks are communicating with each other, but certainly when one starts to bolt, it's creating stimuli in the water, acoustic, as well as near-field particle displacement, which is going to alert the other sharks in the area. And they, even in the dark, when they can't see the other shark bolting, they can feel it, they can hear it, and they're going to respond. So I don't think there's an actual communication per se, but there's certainly transfer of sensory information from one organism to another. Yes, sir, right up front.
Have you seen other sharks trying to hunt the blacktips like bull sharks, great white?
Right. Yeah, so there's certainly other big predators are probably taking out these blacktips. So these blacktips are the same size as me. All right, I'm 5'9". They're about that long. And they're taken out by great hammerheads who we have video. They are taken out by bulls. I don't have that on video yet, but we know from stomach contents that bulls are eating them. Other large sharks are probably also, like tiger sharks, are probably also taking out these blacktips. We don't have that on video. I'd love to get it. If anyone here has a drone and has any cool predation video, do feel free to send it my way. I'd love to see what's going on. All right, yeah, at the back. Why the sharks seem to prefer the colder water, you said? Yeah. Sharks are not like us. Their metabolic rate scales with water temperature. Warmer water, higher metabolic rate. They have to eat a lot more calories in order to get by. Cooler water, they can sort of chill out. They're not burning as many calories to get by. What these sharks do is they spend their summers down here in Florida where the water is the right temperature. As the water continues to warm up in the spring, they start moving their way farther north, and they're spending their summers off the Carolinas, for example. Summer temperature in the Carolina is the same as winter temperature in Florida. They're following that preferred temperature up north, and then as it gets too cold for them up there, they're following it back down south. So they have this thermal preference, let's call it, where they like to be at a certain range, and they will follow that temperature up and down to remain within that range. All right. Let's call it here, and we'll wrap up with more later. I think we're talking to Dr. Grubbs. Thank you.
Okay, for our final presentation, we have Dr. Dean Grubbs, research faculty and Associate Director of Research at Florida State University's Coastal and Marine Lab.
Okay, thank you. Thank you guys for coming. So I took a very different approach to this talk, and rather than talk about one topic, I'm going to try to tell you about 35 topics that are going on in my lab in 20 minutes. So hold on tight. Hopefully the videos work. So I came the farthest. I'm from FSU's Marine Lab up in North Florida, just a little town called St. Teresa, just about an hour south of the main FSU campus in Tallahassee. And so I'm essentially a fisheries ecologist and a conservation ecologist. And so most of the research in my lab goes toward addressing specific questions that are needed for fisheries management or for the conservation of imperiled marine stocks, mostly sharks and rays, but not entirely. And so I concentrate on economically important species, so those that are subject to fisheries, those that are the subject to bycatch overfishing, which I'll talk about a little bit, as well as threatened and endangered species. And so I work with state, federal, and international levels to do this work with a lot of collaborators from academia, as well as NGOs, and as well as governments. And as I said, most of the work's on sharks and rays. So in shark conservation and management, if I think about just the overarching questions that we're trying to address in my lab, they're essentially, at the population level, we're interested in what is the current population status of different sharks. So what are the trends in their abundances? We're interested in life histories. We need to know how long it takes them to reach maturity, how many offspring do they have, how often do they reproduce, those kind of questions. Habitat use. What are the critical habitats for a shark or a ray over the course of its life? What habitat does it need to survive and grow up and reproduce itself? And then community structure. What species make up a marine community, and how do sharks fit into that marine community? What roles do they play? And then finally, I'm not interested in just drawing attention to where there are necessarily problems in declining stocks. I want to know how we can recover those stocks if there is a case when we have endangered species or we have overfished stocks. How do we recover those populations? And a lot of the work recently that I do is also related to bycatch and post-release survivorship of sharks that are caught as bycatch in different fisheries. And so there's an article from the Save Our Seas magazine that I wrote last year on, a couple years ago, I guess, on bycatch mortality in sharks, if you want to download that for free. Because this is probably our biggest and most challenging problem in shark conservation and management is bycatch overfishing. So my work is predominantly in Florida, but also throughout the Bahamas. I do a lot of work on cownose rays in Chesapeake Bay and a little bit of work in Hawaii and some other places. But most of my work is in Florida or the Bahamas, and it's in 3 primary areas. And I'm going to briefly touch on a couple of the projects in each of these areas. We have deep sea ecology projects that are mostly in the Gulf of Mexico, but I'll also end talking about a deep sea shark research project that's in Exuma Sound in the Bahamas. Do a lot of coastal ecology on sharks throughout the Big Bend area. If you don't know, this is arguably the largest contiguous seagrass bed in the world. It goes basically from Apalachicola Bay almost to Tampa. And so this is a big area that we work. There's where my marine lab is. And then small tooth sawfish work as well, and that's primarily in South Florida and in the Bahamas. So when I ask people what is a shark, usually people are thinking about this ugly creature. This is not a great white shark. It's not even maybe it's a mediocre white shark. Official name's white shark. Nothing great about him. It's a fat mako wannabe. It's just, But anyway, people think of that when they think of a shark, that this is a shark. But that's actually a decidedly atypical shark. A typical shark is a little shark about this big that lives in the deep sea. Most species of sharks don't get bigger than a meter, so about 3 feet long. And about half of the living species of sharks in the world live their whole lives in the deep sea, deeper than about 700 feet deep, 200 meters. So that's what a typical shark. These are Florida typical sharks that all live in the deep sea. So I do quite a bit of work in the deep sea. Most of the work in the Gulf of Mexico has surrounded the Deepwater Horizon oil spill. Anybody heard of that? Okay, yeah. So we had the biggest oil spill in the world, basically, right up there in 2010. And so we were interested in how that oil spill affected deep sea animal communities in terms of community structure, the toxicological effects of the oil, did it affect their life histories. And so this led to a lot of different projects on a lot of different taxa. This is actually a giant snake eel, Ophictus rex, so that's not a shark. In case you didn't, it's not a shark. But this species was actually described in the Gulf of Mexico in 1980. This giant snake eel that lives buried in the sediment off the Mississippi River plume. Like, if you ever see the movie Tremors with those things with their head in this dirt, that's how I envision. They're just buried in the sediment with their heads sticking out. This is one of the smallest sharks in the world, the green lantern shark. That's an adult. We caught the only pregnant adults that have ever been seen of that species. That's as big as they get. And then, of course, the six-gill shark, I'll talk a little bit about at the end, is one of the biggest predatory sharks in the deep sea worldwide, actually. We've actually described quite a few new species as part of this project. This is one of the most common species in the deep sea in the Gulf of Mexico. If any of you know who Jeannie Clark was, the shark lady, we named it after Jeannie, so it's Squalus clarkii is what that one is called now. We also had some cool findings in the deep sea, like we caught the first Greenland shark ever caught in the Gulf of Mexico, which is kind of crazy. There's a Greenland shark there. It was caught at about 6,000 feet deep, really close to where the Deepwater Horizon blowout was. And this got a lot of press coverage. I'm not sure it's all that scientifically interesting, but we got a lot of press coverage for it. I even got into Celebrity Bala magazine, which I'm pretty sure this peak of my fame, because I don't think I'll ever be mentioned with Britney Spears again. But anyway, that was fun. But one of the main goals was to look at the toxicological effects of that oil on the deep sea animals. And so these are the three most common sharks in the deep sea in the Gulf of Mexico: Squalus cubensis, Cuban dogfish, Genie's dogfish, and the little gulper shark. And these are basically enzyme activities that are detoxification enzymes. They're basically correlated with the body physiologically trying to get rid of a poison, get rid of a toxin. And so this is in relation to PAHs, the most toxic components in oil. And what I want you to notice is that these three sharks have different depth distributions, that's their depths, but they show amazing concordance across all three of them in terms of when this peaked. They all peaked in toxicological response to the oil about 3 years after the oil spill. So it took 3 years for it to work its way basically up the food chain to these deep water sharks, even though these are little deep water sharks. Well, that's all well and good, but the bigger question is, did it have any effect on their populations or on their reproduction? Did it have a reproductive effect? Did it actually result in mortality? We don't think so, actually. These are, just like Dr. Kajiura showed you his aerial catch rates, basically, his estimates of abundance of the blacktip sharks. These are our actual catch rates in terms of the number of sharks caught on a hook in an hour in the deep sea for those three species. And you can see that if there was a toxicological effect, we might expected there to be a minimum, or at least a steady decline, like we're showing with the Genie's dogfish. This illustrates the need to use multiple species when you're looking at any of these types of effects, because you can see that the other two species actually showed increases across this whole time frame. So we don't think that the oil spill had enough of effect to translate into population level effects in the deep sea. However, I'll end that by saying that actually we saw in some of our other taxa, 7 years after the spill, these levels start to go up again after the oil got resuspended. And of course, that's where all the money ran out, so we don't know what's happening now. We only tracked it for 7 years. Those same three species that I just mentioned, though, it's important to realize that they're not only exposed to oil, they're exposed to fisheries as well. In fact, they are among the most common bycatch species in the deep water snapper and grouper fisheries in the Gulf of Mexico. And we actually we used some caging experiments and blood chemistry to basically try to estimate mortality rates. So I work with my student, Brendan Tower, to estimate not only how many die at the boat, but how many die after you release them. And all of the management plans assume, because they're almost all are alive at the boat, and if you look at the stock assessments and the observer data, they'll tell you that 97% of the Cuban dogfish were released alive. But what happens to them after they're released alive? Well, what we found is that at least 50% of them die within 24 hours. So actually, the mortality rate is at least 50%. And in fact, we found that 55 to 88% of these deep water sharks die post-release. So that's a huge consideration when we start doing stock assessments for these, for basically a stock assessment for grouper and snapper. You have to consider the bycatch mortality on the bycatch species. So we also do a lot of work in the coastal zone. This is basically looking at mostly we're looking at abundance trends over time, and these data are used directly into the stock assessments. We're looking at food webs and community structure, again, life histories. Also, we do a lot of tagging. Actually just had a shark just a couple weeks ago that was just tagged with a dart tag, a sandbar shark I tagged in Virginia in Chesapeake Bay in 1999 or 1998, was just recaptured 21 years later with a dart tag. Pretty cool. It's in North Carolina. Anyway, so this work basically goes from Apalachicola Bay. We're actually extending all the way over here now, almost to Tampa. And we use a spatially balanced random sampling design. So we're not trying to target the sharks. We're letting a computer model just tell us randomly where to go to sample. And we do that every year. We've been doing it for 11 years now, since 2009. We catch about 1,000 sharks in that survey each year from about 12 species. And the point of this is basically we tag and release them all, and we take samples from them. But we're looking at, again, the catch rates, the number of sharks caught on the hooks, that's what this is here, per hour over time. And so these are our data for both Atlantic sharpnose and bonnethead sharks for the last, that includes our data and some other people's data. This is from the last stock assessment for these two species. So this work goes directly into the stock assessments, and you can see that these two are stable. Atlantic sharpnose sharks are probably even increasing. But we're also interested in not only how these shark populations are changing over time and managing them sustainably, but we're also interested in their communities. So how do they fit into the food web? And so a lot of this work involves trophic ecology or feeding ecology, not only looking at diets, but we also use what are called stable isotope analysis. And so you often hear people say sharks are apex predators or sharks are top predators, but are they really? Are sharks really top predators? These are a few tooth morphologies of some of the bigger sharks you might be more familiar with, things like bull sharks and blue sharks and makos and tigers and six-gills. Those are obviously some pretty upper level predators there. These are also shark teeth though, and these are very, very different. So sharks actually probably fill a lot more roles in the marine ecosystem than we give them credit for. And that's where our stable isotope data come in. So these are stable isotope data. Basically, we're looking at ratios of heavy and light nitrogen isotopes and heavy and light carbon isotopes in this standpoint. And basically what this tells us is the higher this ratio of nitrogen, the higher you are on the food chain. So if you're up here, you're top predator. If you're down here, you're lower level predator. The carbon is actually interesting too because it tells us what the primary producer source is, what food web you're in. Are you in a phytoplankton, pelagic food web, or are you in a seagrass-based food web kind of thing? So right there you've got three species of sharks. In this system, the blacktip shark you could argue is the top predator, but you've got a sharp-nosed shark here in the middle, and this bonnethead shark is at least one trophic level below, so it's not a top predator. In fact, this right here, that's a catfish. That's a sail cat. So catfish is higher trophic level than a bonnethead shark. Here's another example using more data, more species from that same region. This is 12 species of sharks and rays from this area. Some of them may not surprise you that the top ones in this system are great hammerhead sharks and bull sharks. Those sharks, as Dr. Kajiura just showed you, they try to eat other sharks. So not surprising that those probably are apex predators in this system. And then you've got blacktip sharks and tiger sharks. Now, it may surprise you that tiger sharks are not a top predator in this system. If you want to know why that is, ask me later and I'll tell you. What will probably surprise you even more, though, is that this little point out here, that's this silly little butterfly ray. The silly little butterfly ray is at the same trophic level or above blacktips and tiger sharks. And that's because it's a fish eater. It's a silly little ray, but it's eating little flounder and stuff. And so it's not quite as straightforward as we often think. Here's another example that kind of drives this home. This kind of surprised us when my colleagues at FWC and I started looking at the food web and the trophic structure of bull sharks and sawfish. And this is actually all the black dots here are sawfish, these guys, over a lifetime from newborn to adult. And these white dots are bull sharks from newborns to adults. And notice they completely overlap in terms of their nitrogen isotopes over from juveniles to adults. So actually, sawfish, this silly ray with this ridiculous mouth and this long hedge trimmer on the end of its head, is at the same food level, same level of the trophic spectrum as a bull shark. And when I started studying sawfish, I was like, they're silly. They just probably eat crabs and stuff. But no, it turns out they're pretty bad predators. I mean, they're awesome. This is actually, that's me pulling a 1 meter shark out of the mouth of a sawfish. So that's a shark this long that the sawfish ate and swallowed whole. That's a serious predator right there. And so that leads me to the sawfish, which is the third major area that we do work in my lab. And so you may or may not know, small-tooth sawfish was the first native marine fish ever listed under the Endangered Species Act. Listed as endangered in 2003. The center of its distribution in the United States is in Florida. We know there are populations in the Bahamas as well, though, and so most of our work has involved looking at if there's any exchange between those two populations. Also trying to figure out the reproductive biology. As is classically the case, the species was overfished and then endangered before we knew anything about its life history. And then we're also monitoring abundance trends to monitor recovery, because sawfish have now been protected for more than 2 decades in the state of Florida. And then we're also looking at bycatch reduction and movements and migration. And so this is the way we study sawfish, just real briefly. This is from a Changing Seas episode that was filmed by the WPBT here. And I don't know why it's all jerky. But we essentially set out 50 hook long lines that are comparable to what a commercial fisher uses, but we only leave them out for exactly 1 hour so that there's no mortality associated with all the sharks, because we catch about, we've caught about 1,500 sharks in the Florida Keys with this method. Then once we get a sawfish, the next question is, what do you do with it? Dr. Burkholder mentioned that they're pretty hard to tag and to deal with. And so you can see they're incredibly strong. They're obviously big animals. I wish it wasn't jerking like that. This one actually almost ended my life right there. I still jerk every time I see that. If you look at my boat, you'll see that there are just holes poked all down the side of the boat from them just knocking holes in the gel coat. I've got a tooth stuck about that deep in the engine of my boat. Once we get them alongside, we count the teeth, we take photos of them, we'll take blood for stress physiology and reproductive hormone analysis. Notice I just drilled a hole through an endangered species. It took me 5 years to get permission to do that. But notice it didn't move at all. That's because there's no blood or nerves or anything in that part of the dorsal fin. So it's actually not very invasive at all. There we're putting on an archival satellite tag like Dr. Burkholder was talking about earlier to look at their movements and see if there's exchange between the U.S. and the Bahamas. And then we take blood from a caudal vein and we use that for reproductive hormone analysis. So the work we've been doing, these are all the places that we've done sets in the Florida Keys. All the little balloons are where we've actually caught a sawfish. And these are just some spot tag, other satellite tag data from Andros in the Bahamas, which is the primary place that we've been doing work there. And most of our work has involved, I think the reason we still have sawfish and the reason we're actually seeing recovery of sawfish now is that the bulk of their habitat is under protection. We actually have most of the sawfish habitat in the United States is either in Everglades National Park or the Florida Keys National Marine Sanctuary, at least for a big chunk of the year. And most of the sawfish habitat in the Bahamas is in Andros Westside National Park. So we have to go to Andros several times a year if we can. So there's all the funders. NOAA, Disney, FWC, Save Our Seas Foundation funds most of our Bahamas work as well. And so we've done 15. I threw this one in here because the field school crew's in the house over here. They're sitting right over there. So we've done 15 of these trips to Andros. Basically, I have to trailer this boat from my lab down to Miami, launch and go to Bimini, clear customs. They come as well and meet us there, and then we tow my boat to Andros, work for a week using the Garvin. This boat here is our mothership. We're waiting to see what this stupid Hurricane Jerry is going to do, because we're supposed to leave in a couple days to go on a trip down there. So hopefully we'll be going back there soon. Some of the big things that we found out about sawfish over the last few years has been that we finally worked out where they're mating. We never knew where they mated. We knew where they pupped. We knew where they gave birth, but not where they mated. But we actually found males and females with fresh mating scars and ultrasound evidence that the females were postpartum, meaning they've already given birth. And so that was kind of cool. And this is all in the Everglades National Park backcountry and in certain backcountry creeks in Florida Keys, in the lower Florida Keys, which is kind of cool. Probably the other coolest thing that we've had happen in this work happened in Andros. And Jake Jerome, who's sitting right over there, he's trying to hide. He filmed this. This is his GoPro footage. This is the biggest sawfish we've caught in the Bahamas. It was 429 centimeters long, so about 15 feet long. And we caught her and we were excited about it. You see she's only in about 6 feet of water, 5 feet of water. And so we basically get her tied up to the boat, let her freak out a little bit, then they calm down. My grad student, Bianca Prohaska, gets in immediately to try to get blood because we want blood immediately. We actually take blood when we catch the animal immediately to see what the stress is associated with being on the line. We actually take blood before we release them too to figure out if we're causing them too much stress as well. But once we flipped her over to tag her, we noticed something weird happening. I don't know, there's some kind of movement going on in the thingy there. And so that's actually 2 rostra of baby sawfish right next to each other, trying to get out at the same time. So I'm the only person who's ever played midwife to a sawfish, and I got to actually pull these babies out. We didn't try to take any out that weren't coming out on their own. There were more inside of her I could feel moving around in there. We pulled those first 2 out. They were trying to get out at the same time. We were worried they would get damaged while the mom was there when we needed to do surgery to put a transmitter in her. And a third one started coming out. We pulled that one out as well. There you can see the size of a newborn sawfish versus mom. You can see they have this gelatinous sheath around the rostrum, which protects the teeth and probably also protects mom. I mean, giving birth to this thing full of teeth, that's not very pleasant. And so then we flipped her over to, we put pit tags like you put in your dog or your cat and the babies. And then we were getting ready to tag mom and implant a transmitter in her. There were 3 at this time. There's Jake. He sets up his GoPro. Mom freaks out and knocks my colleague Andrea Krutz over. And when she did that, she squirted 2 out on her own. And so she just birthed them on her own. Brian caught one and handed it to Jake. And then you'll see they're all looking for the other one. It's kind of disappeared in the murk and everything. But then in a second, you'll see it'll come swimming through. And I'm glad you're here, Jake, because Jake's expression is one of my favorite things here in a second. Here comes a little sawfish coming up. My student, Brian Keller, just bare hands. It's like, oh, hey. Nick grabs it, and Nick says, here, Jake, have 2 sawfish. And so Jake is probably the first person ever to hold 2 baby sawfish in the wild in the world. And so, he's just, they had never seen a sawfish before this day, and so this is amazing. And so this is one of the releases of the little babies. They swam off. They were the same size as the ones that we catch in the wild in Florida. They have a yolk sac that they use for nourishment as an embryo. That yolk sac was completely gone. That means they were ready to be born. So that was pretty cool. A cool thing. So Dr. Burkholder talked about some of the technologies that we have at our disposal now. One of the really cool things now in acoustic telemetry instead of satellite telemetry is that now we have lots and lots of researchers working together and putting out these huge arrays of receivers in the Gulf and in the Atlantic. And so now we can actually implant acoustic transmitters that will have a battery life of 10 years inside mom. And so if or inside any of these sawfish. And so if they go by any of these receivers, we're going to know about it. So this is how we're going to figure out if there's exchange between the U.S. and the Bahamas. Spoiler alert, there's not. It's separate populations. But it's provided us with some cool data. I'm just going to show you one example of an animal that did some cool stuff. This is a 383 centimeter male that was tagged off of this wreck, the Queen of Nassau. If you know Rob Stewart, the guy from Sharkwater who died in the Keys, this is the wreck he died on. He was there because I had told him there were sawfish there. So we tagged this animal there on the April 1st of 2017. Then it went up in April 23rd, 2017. It was up off of Cape Canaveral. We didn't even know they really went up off of Cape Canaveral. Stayed up there for a few weeks, and then between May 6th and May 29th, it went all the way back down to the lower Keys. Then it decided, well, the Atlantic was okay. Let's try the Gulf. It went all the way up off Tampa, way offshore off Tampa. Didn't like the Gulf. Came back down, and it hung around in the lower Keys for quite a while. And then it hung around in the Keys and in Florida Bay, and even went through Miami briefly to do some salsa dancing. And then it went back up to Cape Canaveral on the same day that it showed up on that receiver the year before. It was on that same receiver. Stayed up there for a little while, and then it came back down to this area. It was down in the lower Keys from October 2018 to February 2019. And then, believe it or not, 4 days prior to the other 2 years, it showed up on those receivers again. Crazy the amount of data we're able to get from one animal now using this technology. But That's all cool, but what we're really using this for is to look at bycatch risk and mortality risk in these populations and their migration patterns. And so we've had these animals detected on 350 receivers from Appalachia. These are all animals tagged in the Lower Keys and Florida Bay. And they've been detected as far north in the Gulf as Apalachicola Bay and all the way up to Georgia in the Atlantic side. And so we're tying those data in with satellite tag data. These are similar to the mako shark data Dr. Burkholder showed, but except that these are much more constricted. They don't travel nearly as much as those makos do. And we're able to take those data together, overlap them with fishing effort, and figure out which fisheries are most likely to catch sawfish. And this is preliminary, but this shows us that the gillnet, offshore gillnet fishery off of Cape Canaveral may be of major concern if they overlap temporally. Longline fishing risk is mostly down in the Lower Keys. Shrimp trawl risk is mostly in the Dry Tortugas area and then a little bit more up in the Charlotte Harbor region as well. And so then we can look at those fisheries, figure out when they're overlapping with sawfish, and hopefully put in some mitigation measures to prevent overfishing. So the longline fishing we're not too worried about. This is a paper we published, my student Janka Brohaska, looking at stress physiology. Lactate is an estimate of your physiological stress. It's a signal of blood acidosis associated with stressful exercise or whatever. And these are data from a whole bunch of sharks, and you can see what levels they're at. And these are all sawfish here, including those caught on longline and in gill nets. And you can see it's quite low. They're pretty tough animals. They're quite resilient. But when they get caught in a shrimp trawl, that's game over most of the time. So the shrimp trawl risk is really what we're trying to mitigate for. At the opposite end of the spectrum, ones that are not very physiologically resilient are the hammerheads, great hammerheads and scalloped hammerheads. So we're also looking at the effects of capture on great hammerheads and scalloped hammerheads, particularly in longline fisheries. A sawfish gets caught on a long line, it lays on the bottom and it breathes and it gathers its strength and it gets ticked off and it waits for you to bring him to the boat and it tries to kill everybody. And then you release it and it's fine. A hammerhead, not the case. They're very fragile animals. And so we've been doing the same kind of work using satellite telemetry combined with stress physiology data and actually measuring the time the animals are on the hook to look at their likelihood that they're going to survive. And basically what we've found is that once they're on the hook for about an hour, both species, their stress level tends to go up. But it goes up much quicker in the great hammerhead than it does in the scalloped hammerhead. And actually that's what we're finding with the survivorship tags too, is that whereas the scalloped hammerheads will survive several hours on the line and you're releasing them, the great hammerheads do not. And so the reason we're doing this is there was a question whether maybe just decreasing the number of hours a longliners' hooks are allowed to be in the water would decrease mortality. Not really, because they're going to set, it's only logistically feasible to set at 4 or 6 hour soaks. And so that's too long. So what that means, we're going to have to shift instead to probably time area closures, basically closing areas where the hammerheads, we know they are, and not allowing longline fishing in those zones. And the fishermen are open to that, and they're actually doing a lot of that on their own already. And so the last thing I'll mention briefly, no, I'm going over, related to this post-release survival work, as I mentioned, we started doing some of this with deep water sharks, including those 3 small species in the Gulf of Mexico. We've also been interested for years in the post-release survival of the big animals. You know, if you take a big 6-gill shark and you bring it on the deck of a ship, would it survive? Now, you've all heard of finning, as Dr. Burkholder mentioned to you, and someone mentioned cosmetics as a use of sharks earlier. There's actually a deep sea equivalency to finning that we've termed livering, which is often deep sea sharks are caught as bycatch in these deep fisheries, and the meat's not marketable except in Europe, really. And so the liver is removed because it's valuable, and the rest is thrown overboard. And one of the rationalizations is the shark was going to die anyway. So we might as well just take what we can sell. So we started asking the question, is that true? Would the shark die anyway? And so we started this work looking at the effect of bringing a big 6-gill shark on the boat, tagging it and everything, and releasing it and seeing if it would survive or not. And what we found, we've tagged about 25 of these things in 5 different regions around the world, Here's off Virginia, the Gulf of Mexico, the Bahamas, Hawaii. Across the board, they survived. The only ones that didn't survive were ones that got eaten, and I can't help that. But they got eaten, and if you want to know how they got, I know they got eaten, ask me later, and I can tell you how we know that. We had 2 that got eaten by other 6-gill sharks. We had 1 that got eaten by a tiger shark, and if you want to know how I know, ask me later. And we have a tiger shark that we tagged that got eaten by a 6-gill shark. So it's all it's rough down there, for a big shark. So, but across the board, these things survive. But what we noticed is that the first 2 days or so was very different everywhere. They sort of had a freak out period. So it seemed like there's definitely some sort of stress associated with capture, even if they survive. Of course, how would you possibly tell if that's what's happening, if this is actually short-term stress associated with capture? Well, the answer is you tag them without catching them, right? Well, how do you tag an animal that lives 2,000 feet deep without catching it? Well, you convince somebody to let you strap spear guns to a submarine and go tag sharks. I don't know how we managed to do it, but we did. With Ocean X, we managed to convince them to let us strap 2 spear guns with satellite tags attached to them on submarines, and we went down, and we basically went down to 2,000 feet deep. We strapped about 500 pounds of bait on the front of the sub, and we just sat there in the pitch dark until, looking through a little red headlamp, until we saw something that made us think there's an animal there. The first dive I did was like about a year ago, and we sat down, and there's like a sheer rock wall with silt, and we're sitting in the dark looking through an ultra low light camera, and all of a sudden there was dust everywhere, and we, including the sub operator, thought we were about to be killed because there were rocks tumbling down off of this cliff. We turned on the lights, and it wasn't. The silt was from a 6-gill shark. Its tail had brushed the side of the wall and just kicked silt in everywhere. All right, we didn't die. And so then we turned the lights off again and just sit there in the dark. And so if this next video works, you'll see what we saw when we're sitting in the dark, and all of a sudden the ultra low light camera went black. We'll see if this actually works. Fingers crossed. And so we're sitting in the dark. We're sitting in the dark. We're still sitting in the dark. Yeah, we're still sitting in the dark. I don't think it's going to work. What we saw was that all of a sudden we flipped it on. This giant 6-gill shark was right up, had the spear in his mouth, and was right up against the submarine. We'll see if this one works. That one's not going to work either. That's okay. And so you can actually download and look at the videos online. You got a lot of press coverage recently, and there's going to be a documentary coming out soon. But this is the first 6-gill that we saw. I actually took this picture with my phone from inside the submarine. It's kind of cool. But we actually, if you go to National Geographic, has an article online, you can watch a really cool video of this giant 6-gill that we did actually finally tag using a submarine. First time ever that's happened. This was and there's some really cool video of this giant 6-gill just pushing the submarine around and everything. It's really crazy. The tag's on for 3 months. It should be popping off any day. And we haven't heard from it. That's a good sign. That means it's still on there. And so we're waiting to hear from it. And I think that's it. I'll end there. Yeah. And so that's it. One or two quick questions before we go up to Jeff.
You were saying that the tiger shark, you were going to tell us why the tiger shark wasn't on the top of it.
Oh, why tiger sharks aren't on the top of aren't really apex predator. Anybody know what tiger sharks like to eat besides everything? Turtles. What do turtles eat? Seagrass and sponges and stuff. Turtles are actually essentially very low level consumers. So if you have a tiger shark eating, cow, it's going to have a relatively low level. Whereas if you have a bull shark eating a shark, it's eating fish, then you're going to be way up higher. That's why tiger sharks are low.
You said 6-gill shark got eaten by a tiger shark. Tiger shark got eaten by a 6-gill. Which shark is more dangerous to the other tiger shark? 6-gill or 6-gill?
So I don't think either one is more dangerous to the together, they probably don't overlap all that often. Six gills live at, no matter where you are in the world, they basically spend, sorry, I like to walk around. They spend their days down at about 5 degrees Celsius and their nights up at about 16 degrees Celsius, okay? So in the Gulf of Mexico, you're talking, they're up at about 200 meters during the night, and they're down at 800 meters during the day. Where we were in the Bahamas, you're more like 400 meters at night, 1,200 meters during the day. Tiger sharks spend most of their time relatively shallow, but then they make these bounce dives down to 400, 500, 600 meters. They don't seem to spend a whole lot of time down really deep, but I don't know, of the 500 tiger sharks I've ever caught, probably 80% of the ones that I've caught over 13 feet were in deep water. But I don't think they spend that much time, so they probably don't overlap all that often. But they probably follow our boats and wait for us to release sharks. That's another thing.
You were talking about mortality, about bringing these deep-water animals up to the surface.
Yeah.
What kind of physiological shock are they going through because of the amount of temperature change and pressure change and everything else like that? Is that affecting them as well as when they're probably not in the water and not breathing for a while?
Yeah, so great question. So that's what we're trying to figure out, is what actually causes the mortality. I think for the small ones, the small sharks that we're seeing, the high mortality, I think is thermal stress. They're animals that live down less than 50 degrees Fahrenheit, and you're bringing them up to 80 degrees Fahrenheit, and I think they cook, and so they die quickly. The big sharks like this, though, we put thermistors in them and released them and shown that they actually have such thermal inertia, because they're such big, bulky animals, that it takes them 8, 10, 12 hours to warm up or to cool down. And so the temperature stress probably isn't much of an issue. We thought light stress might be an issue for them, because they're meant to, I mean, they're really adapted for really dim blue-green light. But we tagged them in the day and tagged them there at night, so there's no difference in their behavior. The pressure, they don't have a swim bladder, of course, so they don't have barotrauma like a bony fish would, like a grouper or something. But there are changes in enzyme activity and oils in the liver with that pressure change. So pressure differences could play a role, especially in some of the really big liver deep sea sharks like the gulpers and things. So pressure is definitely an issue too. Oxygen, they're pretty tough. They don't seem to, I don't think that has much of an effect. They're living in a relatively low oxygen environment anyways. But we're still trying to figure that out.
I think what we'll do, for the sake of time right now, is I'll invite all of the researchers up to the stage. If you have questions for an individual presenter, maybe we can talk them into sticking around for a little bit at the end and answer questions then. So if I could invite the presenters to come up to the stage, we'll just do a brief Q&A session. I have my list of questions, but I'll let you guys ask yours as well. So come on up. We might be sharing microphones. Okay, does anybody from the crowd have a question for all of our presenters here? Otherwise, I will get us started a little bit. Yes. Can you...
There's been a lot of controversy about when you tag like a big animal, like great white when it's a juvenile, and then you see pictures of it when the dorsal fin is like this. Has there been any changes in behavior when you put that kind of a tag into the shark? Does it change the behavior? Does it do anything harmful to the shark?
Thank you.
So it's a good question. So we've actually had debates within our community over proper tagging techniques, and I will say not everybody follows them. And so there are ways you can minimize the damage done. If you're seeing an animal that's not behaving what you perceive as being normally, then it's probably not tagged properly. We've seen animals that were not tagged properly, and there's severe necrosis of the dorsal fin and things like that associated with it. So we try our best to avoid that. I wish I had thrown it in there. I have a picture of that's actually one of the reasons why I started using the drill and putting this harness, so that there's not much making contact with the animal from my satellite tags. And so with those sawfish, I actually had one recaptured a year after the tag released off of it, and you would never know it had been tagged. There's nothing there, and that's the goal. So I think we need to do better about making sure everybody's using the right techniques.
Yes.
Do you have a mystery shark that you would really like to know more but can't get more information on, like collectively or individually?
I do. So the one I really want to study is the winghead shark. This is the hammerhead with the biggest head. The width of the head can be half the length of the body. So you have a shark that's 1 meter long, the head is 50 centimeters wide. I've twice been skunked in my attempts to get this shark. I was funded by National Science Foundation to go up to northern Australia to catch them. One time we were blown out by a cyclone. Another time we got there right at the end of the season, and my guide caught a shark the day after I left. So I have been frustrated by this winghead shark for years. I need to see that one.
The only winghead shark I've ever seen was one that Steve got that came to when we were both in Hawaii, and it was going to some museum, right? Yeah. And you and I took about 7,000 photos of this dead shark.
Yes.
As a collective, we're heard that about 100 million sharks are being caught and killed every year, and that they range through a large number of trophic levels from apex predator all the way down to pretty low levels. So, have you seen any effects on general ecosystems directly attributable to the loss of sharks? Similar to the keystone, not keystone, the loss of the wolves in the national parks. Have you seen anything like that going on a large scale?
So when I first started teaching shark biology courses about 100 years ago, I used to use the lions of the Serengeti, wolves of Yellowstone analogy for top-down effects. The truth is there's very little robust evidence that occurs in most marine systems, and part of the reason is exactly what I was getting at with the food web stuff, is that you often have in marine systems functional redundancy where you don't just have one apex predator, two mid-level predators, and then 2 or 3 below that. It's not a linear sort of system. And so Bob Payne from the University of Washington, who passed away a couple years ago, he's the one who coined the term trophic cascade and keystone species, actually. He was on our science advisory board at our lab. And he and I used to debate this a lot, because the system that he described that in is rocky intertidal communities, where you have the apex predator is a sea star, and there's no mistaking the effects of removal of the apex predator. So there are probably some systems where we could see it. I think on certain coral reefs you could see that you can see this happening, because you do have a relatively confined food web. But I don't want to get, these guys know I'll go on a tangent. We get on, there's the whole idea about the shark-mediated trophic cascade in the U.S. that caused the release of cow nose rays, which caused the cow nose rays to eat all the oysters and scallops. Well, that was wrong on every level. And we published a rebuttal showing it was wrong at every level. But we have to be careful and make sure that the science is solid if we make such claims, because the byproduct of those claims was that now there's a fishery for cow nose rays trying to reduce this hyperabundant mid-level predator, but when in reality cow nose rays produce 1 pup and have a gestation period of 12 years. They're one of the most vulnerable sharks or rays we have, and so they cannot sustain a fishery. And so, to answer your question, there's very little direct evidence. I'm sure it happens in certain ecosystems, but for a lot of people, that's sort of been unfortunately, a lot of people, instead of approaching ecosystem research as, let's figure out how this thing functions, whether it's top up, bottom down, middle out, whatever it is. They start trying to look specifically for this top down effect. And it may not be there, you know.
I was just going to go off of that. Yeah, so a lot of what we know about these keystone species and some of these really important ecological systems are terrestrial because we can observe them and we can study them a lot better. And if you remember that, like, 70% of this planet is water, right? What we can see and what we know is really limited in terms of what we know compared to terrestrial ecology or even intertidal ecology that we can see and study a little better. So, yeah, there's a lot of redundancy in systems, and we just don't know a lot. Like, we're tracking sharks because we don't even know where they go. Like, you can't study the effects on the ecosystem if you don't even know, like, where they're mating. What are they doing these things? Like, you can't simply know that information because we have so much that we still need to learn.
Yeah, and I'll go a little off that as well. As they're saying, that everything is so interconnected that there's a lot of different levels that these are all working together. There has been a lot of work done in Shark Bay in Western Australia that does show that there is likely some amount of top-down effects in some of these systems. There, the tiger shark, it impacts most of the other species that are there. It's eating the dugongs and the turtles and the dolphins and the sea snakes and the birds and the fish. And so a lot of that redundancy goes away because they are impacting at all of these different levels. And so they're, much of the work of Mike Heithaus and some of my dissertation work and things like that as well has shown that there is some amount of this top-down effect there. But it is all so interconnected that in many places it does seem to get washed out a little bit as well.
In my opinion, and there are certainly people who will disagree with me, but I think the best thing we can do is to promote and push up the sustainable fisheries that are actually making full use of the animals and have stable populations, and use those as sort of the poster fisheries. And perhaps even have some countries that are willing to do so certify fisheries as sustainable or not. Only take fins that are sustainably harvested. Now that's difficult because Trinidad, for example, is one of the largest fin trade countries in the world now. And Trinidad doesn't catch all that many sharks themselves. They come from other places. And so it's difficult because the fins come from all over the world. And so it's not an easy solution. But if we could actually certify fisheries that are sustainably harvesting meat and fins and everything else and separate those and not purchase the ones that are illegally caught through finning, I think that would be one huge step towards sustainability.
There's just one thing I'd add to that. It may be naive and maybe pie in the sky, but I think education is going to be the key. You're going to need to convince these youngsters out there that going forward, this is not going to work. Don't eat the shark fin soup. And I don't think you're going to convince the elders. Their mind is set. They know that that's what they're going to eat at their next wedding or banquet or whatever. But if you get the next generation and start convincing them of the value of these animals as living entities rather than in a bowl of soup, I think that might be the way forward sustainably in the future, is to change the mindset.
That's a great point that Dr. Kajiura just brought up, because that's actually happening already. We're seeing that already. The demand for shark fin soup is going down. And actually in our own U.S. shark fisheries, back in the peak of the fishery, when we opened trade with China in '79 and our fishery really took off in the early mid-1980s, and it became overfished by about 1992 severely. They were getting, to the fishermen, $20 a wet pound for the fins and 25 cents a pound for the meat. Thanks to more than 2 decades of aggressive management, most of our stocks are now sustainably managed. There are some issues, dusky sharks, hammerhead sharks, but most of them are sustainably managed. But now we're at the point where actually the fishermen, it's a tiny fishery compared to what it once was, and it has to be. Every shark fishery could be managed sustainably, but it may be at a very low level. We're currently managing our East Coast shark fishery at 8% of their peak landings. That's the sustainable level, 8%. But they're actually getting as much for the meat or more for the meat than they are for the fins now. And that's
And this also has to do with food, right? And so the more we start to understand climate change and start to try to mitigate these things, people are trying to eat themselves at lower trophic levels, much like some of the rays and that tiger shark that only eats the turtles, I don't know. But, we're trying to, as humans on this planet, do things that make our lives more sustainable and try to think about that in eating at smaller and lower trophic levels. And so that is also necessarily going to have impacts on the animals that we are currently overfishing and not sustainably using as we start to become more aware of that and make those choices going forward, thinking about climate change and larger societal issues, I guess. If no one eats sharks anymore, there will probably be more in the ocean. That's my hypothesis.
I have one more question. Each one of you, when people say, why are sharks important, what do you say?
You start.
Well, for me, they're an important model for me to understand how skeletal systems evolved, right? Because sharks have maintained their body shape and their cartilaginous skeleton for 450 million years. So there's a lot that I can learn as a biologist looking back to them as far as evolution of skeletons, how we all evolved, and then what I do as a biomechanist is I draw inspiration from them and try to be able to make things that are more fuel efficient, things that are better at maneuverable, and things that we can use to actually make things better now. So they're a great model for me in that respect, and I like them.
Well, I agree. I also like them. That helps. But for me, I think it's one of these animals that people see and they get excited about, and I think it's a great way for us to educate about the oceans and about many different parts of the oceans. I do talk a little bit, usually when asked that question, about sort of their ecosystem role, and that they are a big part of these ecosystems and kind of helping balance these ecosystems. But I think one of the big attributes that they do have is that they are one of those charismatic megafauna, right? Those things that everybody like to see, they like to learn about, and people can connect with. And I think if you can connect with something, that then you can start to get excited about it, get passionate about it, and that will, kind of flow out to everything else. So that's one of the big roles that I think they play, both from the ecological role that they're playing, but also with connecting people to our oceans.
I guess since I built my whole lab on sharks and rays, if there were no sharks, I'd be unemployed. So I have a vested interest in being very actively involved in sharks. And I think it's the exact same thing that Dr. Burkholder said. They are charismatic. They give you the hook. People are interested, and once you catch their interest with the sharks and the rays, then you can pivot and talk about their ecological importance. This is why they're important. They perform this vital role in the ecosystem, and you're able to bring it into the bigger picture and show how they relate to the whole marine ecosystem, not just focus on the sharks themselves, but use them as a way to get people's attention. So I think that's probably one of the things that we all do to use this to get people to talk, and then feed them some real science in between.
So as I tried to point out earlier, sharks fill many different roles in marine communities. And so we often want to think that something has to have a really strong direct effect for it to be important. But even if there aren't always strong top-down controlling effects, that doesn't mean they're not important. They're still a critical link in this web. And so properly functioning marine ecosystems have to have sharks and rays in them. They have to have everything else in them too, but they have to have sharks and rays as part of that. So they're a critical part of marine ecosystems. They're also critical to biodiversity. This is an evolutionarily unique lineage of vertebrates. And so from that standpoint, from a biodiversity standpoint, they're critical. I point that out with the sawfish because there's only 5 species of sawfish in the world, and they're all critically endangered. If we lose them all, we've lost an entire family of some of the biggest shallow water tropical predators on the planet.
I think that's a great question to end on. It is 8:15, so I want to respect everyone's time. Are you available to stick around for a few minutes? If you have additional questions, our presenters will stick around. So thank you for joining us, and please check out the website for our future events. Have a great evening.